Hey everyone, Dr. D here, and in this video we are doing exam 3 review from our Talaro's Foundations in Microbiology 12th edition. This exam review will cover chapters 14, 15, 18, and 19. So let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
All right, let's get started with chapter 14. Remember, this chapter covers the branch of the immune system called the innate or natural immunity. Obviously, you should know the difference between innate immunity and adaptive immunity. You should know that with innate immunity, it's present at birth. It provides non-specific resistance. It doesn't form a memory. Whereas adaptive immunity is specific and it must be acquired and it does form a memory. And then, yes, you should know the 3 lines of defense. So for instance, you should know that the first line of defense is part of the innate immune system. You should know that the examples are right here in this box on the right. You should know about the second line of defense. It's part of the innate immune system. And you should know that it includes these examples on the right. And the same thing with the third line of defense. You should know it's part of the acquired or specific immune system. And you should know that it includes the B and the T lymphocytes. And then do you remember the first line of defense, how it includes all the different structures here listed? And again, as always, remember that the concept checks may appear on the exam. So it's worth learning them and remembering them. So remember this one. Which of the following is not an example of the immune system's first line of defense? And do you remember the answer? It was inflammation, right? That's the second line of defense. You should definitely know about the white blood cells, how they're called leukocytes. You should know the difference between white blood cells and non-white blood cells. Remember, the erythrocytes are the red blood cells, and then the platelets, which are also not white blood cells as well. You should know that these patterns that are found on pathogens called PAMPs, or pathogen-associated patterns, these patterns are recognized by receptors on the white blood cells called pathogen recognition receptors. And then we talked about hematopoiesis. Do you remember that there are hematopoietic stem cells in the bone marrow? And those are the cells that differentiate to give rise to all of the blood cells. Remember, platelets, red blood cells or erythrocytes, and the leukocytes, the white blood cells. And then do you remember how we went through and we characterized each blood cell and we defined what they do, right? We talked about what they do. So you should remember that. So for instance, you should know about the neutrophils. You should know that these are phagocytes, right? Phagocytic white blood cells. But here's something important you should know about neutrophils. And let's see if you can remember better than Wicket. All right, let's quiz you real quick. Is a neutrophil a professional antigen presenting cell? Yes or no? That's right. Wicket is always no. Neutrophils are phagocytic, but they are not one of the professional antigen presenting cells. So they do not possess MHC1, which means they cannot present foreign antigen to helper T cells, you know, those CD4 positive T cells, to activate them. And then you should know about the eosinophils. You should know that these are the ones tasked with destroying eukaryotic pathogens. And remember, that means helminths and protozoa mainly. And then you have the basophils. You should know that these release chemical mediators, mainly histamine. And you should know that the mast cells also release histamine. So that's what they do. Next, remember the lymphocytes are a type of leukocyte. There's the B lymphocyte or B cell and the T lymphocyte or T cell. And these are Oh, hi, Punks. Look, everyone, it's Punky. She's grown quite a bit already, you can tell. Look, you guys, it's Punky. She's growing every single day. Yeah, she's come to say hi. Hi, Punks. All right, Punky, you go play. What a sweet cat. So yeah, we have T cells and B cells. And do you remember B cells and T cells are both formed in the bone marrow? But remember this, B cells mature in the bone marrow, whereas T cells mature in the thymus. Don't forget that. And then don't forget about the monocytes. Monocytes are phagocytic white blood cells, but they only hang out for a few hours before they change or differentiate into macrophages or dendritic cells. You remember the macrophages and the dendritic cells are phagocytic, and they are also antigen presenting cells, professional antigen presenting cells, which means what? It means that they possess MHC2, right? And they can activate helper T cells. And do you guys remember the third professional antigen presenting cell? Can you beat Wicket? That's right, Wicket. B cells. B cells are also professional antigen presenting cells, and they are technically phagocytic cells as well. So if you're keeping track, let's list all 5 phagocytic cells. How about that? How about the macrophages, the dendritic cells, the neutrophils, the B cells, and the monocytes And remember, eosinophils have a minor phagocytic role as well. But of all those phagocytes, what were the only 3 antigen presenting cells? Remember, they were the macrophages, dendritic cells, and the B cells, right? So remember that as well. And then do you remember the natural killer cells? Those were cells that could destroy your own nucleated cells, your own cells, if they're in distress, right? Do you remember if they internalize MHC1? Any cell that internalizes MHC1, signals itself to be destroyed by natural killer cells. And do you remember the only 2 non-white blood cells that are formed in the bone marrow? The erythrocytes, which are the red blood cells, and the platelets, which are involved in clotting. Remember, the red blood cells are involved in gas exchange, oxygen delivery and CO2 evacuation. And then the platelets are involved in blood clotting. Yes, oh, you're so sweet. She's purring and mewing. Okay, we're just gonna take a nap probably soon. Okay, let's see. Oh, and do you remember the lymphatic system? Do you remember that? You should know this. You should know that the lymphatics, the lymph fluid flows only towards the heart, not away, right? You should know a few things about the lymphatics, right? The lymphatic system. You should know that it's the lymph fluid flows only towards the heart. You should know that the lymph fluid is not blood, but it's a type of extrastitial fluid, the fluid that leaves the blood vessels. Yeah, see, lymph fluid is not blood. It is a plasma-like liquid carried by the lymphatic circulation. It's formed when blood components move out of the blood vessels into the extracellular spaces. And do you remember how the lymphatics move? You know how the lymph fluid moves through the lymphatic vessels? It's not because of the beating of your heart. Your heart does not move the lymph fluid through your body. It's the contraction of your skeletal muscles. When you contract your muscles, that's what moves that fluid towards the heart. And remember, it's unidirectional flow towards the heart. And once it gets near the heart, remember, it dumps into a major vein returning to the bloodstream. Next, you should know which are the primary lymphoid organs. You should know that that includes the thymus and the bone marrow. You should know that for sure. You should also know about the spleen, how it's a type of secondary lymphoid organ. You should know that the spleen, not only does it filter out blood, but it also is a meeting place for white blood cells like the B cells and T cells and pathogens or foreign antigens in the blood. So it has two different roles, and one of those roles is in your immunity. And again, do you guys remember, where do T cells mature? In the thymus. So you should definitely know that. And definitely know inflammatory response. Do you all remember the 4 signs or symptoms of inflammation? Redness, warmth, swelling, and pain. These are the Latin terms associated with, redness is rubor, warmth is calor, swelling is tumor, and pain is dolor. So not only should you remember the English, but also the Latin terms, because they're still often used. And then do you remember that during inflammation, these neutrophils migrate to the site of infection? Do you remember that? So you should know these terms. One is margination. Remember, margination was the sticking of the margination was when these neutrophils stick to the inside of the blood vessel. And then you should know the term diapedesis. Diapedesis is when the white blood cells squeeze through the blood vessel out to where the injury is in that interstitial fluid. So again, know the terms margination, diapedesis, and then chemotaxis, which is directed cell movement. Learn the term pyrogen. Pyrogens are substances that induce fever. And do you recall that there are some benefits to fever? Again, this is a list of the phagocytic white blood cells. But remember, of these, only the macrophages, dendritic cells, and the B cells are also the professional antigen-presenting cells. Also, you should learn the steps here. You see these steps involved in phagocytosis. And then you should remember interferon response. Remember that interferon response is a major way our body combats viral infections. One thing you should know about interferon response that you should remember for the exam is that the adjacent cell, the cell that gets the interferon signal, it can still get infected by virus, but it can't be taken over by virus. Does that make sense? Because it's built these antiviral proteins that degrade viral DNA or RNA. So what you should know again is that interferon response is good. It signals, the interferon signals to adjacent cells. Those cells can still get infected. They just can't become factories. They can't become viral factories, and viral replication cycle does not complete. So essentially, the buck stops here. The viral infection stops here. And then, do you remember I told you about complement proteins, how they consist of 30 or so blood proteins or serum proteins, and they get activated and they have all these different jobs to do? You should know this. These are the 3 main jobs I want you to know about. Complement can form a membrane attack complex, or MAC, which is essentially a pore that pokes holes in the bacteria. It also can form opsonization, which means that the complement protein can stick to the pathogen, coating it for phagocytosis. So you should learn this term opsonization. Anytime something sticks to a pathogen and then flags it for phagocytosis, that's called opsonization. And also recruitment of inflammatory cells and cytokine release. Remember, this is complement protein 3,5A. So I do want you to know what the complement proteins do. So this, you should learn this. For instance, you should know that complement proteins C5b, C6, C7, C8, and several C9s make up MAC, whereas complement protein 3b is the one that opsonizes. And then you should know about complement protein 5a. That's the one that plays a role in recruiting inflammatory cells. So basically causing chemotaxis of these inflammatory cells or phagocytic cells. And then this last concept check here from chapter 14, we can wrap up chapter 14. Which of the following defenses is most likely to be active during a viral infection? That's interferon. Okay, so that's it for this chapter 14. Let's go on to chapter 15 and continue on with our review. All right, let's carry on with chapter 15. Remember, this is the adaptive or acquired immune system. It involves mainly the B cells and the T cells. Remember that each B cell and each T cell is very different and very specific towards a particular antigen. And remember that these are the cells that once they become activated, they can proliferate and differentiate, forming memory cells whose job it is to linger in the lymphatics and remember that antigen for future infections. This is why you become immune. And again, do you remember B cells and T cells come from the bone marrow, but the B cells mature in the bone marrow while the T cells mature in the thymus. And then after that, they migrate to lymph nodes and things like this. And then do you recall the antigen presenting cells? These are the B cells, these are the macrophages and the dendritic cells. And remember, just because antigen presenting cells possess MHC2 doesn't mean they don't also possess MHC1. They can also possess MHC1 because all of your nucleated cells possess MHC1, but it's the professional APCs that also possess MHC2. So again, do you remember MHC class 1 is found on all nucleated cells? MHC class 2 is only on those professional APCs, the B cells, the macrophages, and the dendritic cells. Do you remember this? This is so important for the exam. Foreign antigen presented on MHC1 activates which lymphocyte? Can you beat Wicket? That's right, Wicket as always. It's the cytotoxic T cells or CD8 positive T cells. Those are the ones tasked with identifying foreign antigen presented on MHC1. And then those are the ones that become activated and destroy cells that are producing that antigen, right? Presenting that antigen. So that's how cytotoxic T cells work. They recognize foreign antigen on MHC1, and then they go and become activated, in which case they proliferate and differentiate, and then they go on and destroy any of your cells that are also presenting that particular antigen. And then let's see if you can beat Wicket again. MHC class 2, like foreign antigen on MHC class 2 activates which of the lymphocytes? Can you beat Wicket? That's right, Wicket. Those are the helper T cells, the CD4 positive T cells. They're the ones that get activated by recognizing foreign antigen on MHC class 2. And remember, when those helper T cells get activated, they also proliferate and differentiate, forming those effector helper T cells as well as memory helper T cells. And let me ask you this. Who do the activated helper T cells help become activated? Who do the helpers help? Who do activated helper T cells help? That's right, the B cells, right? The B cells. Without help from an activated helper T cell, B cells typically don't become fully activated. And please remember that all of the lymphocytes have different receptor specificities, right? So two B cells are not going to recognize the same exact antigen or epitope on an antigen. Two different T cells, because each has a specific structure on its antibody or its T cell receptor. So you should definitely know that all lymphocytes do not share the same specificity for antigen. And do you remember how B cells work, by the way? B cells, they recognize foreign antigen using their antibodies, which are stuck to their surface. Is called the B cell receptor. They phagocytose that antigen, they process it, and then they become activated once a helper T cell also recognizes that antigen. It's presenting on MHC2. And then, do you remember the B cells, once they become activated, they also proliferate and differentiate, and they form memory B cells for later, but they also form plasma cells. So you should know that B cells, activated B cells, become plasma cells. And plasma cells release tons and tons of antibodies into the blood. Remember that during clonal deletion, this occurs, during maturation of the B cells or T cells. Remember, this is where any B cell or T cell that recognizes self antigens is eliminated via apoptosis. And again, look at this concept check. We already covered this. All lymphocytes have the same receptor specificity. That is 100% false. In fact, each one has a different receptor specificity. Next, you should know about antibodies. Do you remember that every antibody is 4 proteins? Remember, 2 heavy chain proteins and 2 light chain proteins? So you should know that antibodies are made up of 4 proteins. And do you remember, it's the B cells that have these antibodies on their surface, right? Those naive B cells have antibodies on their surface. It's also known as the B cell receptor. And do you recall that antibodies have a variable region at the tip of the Y and then a constant region at the base? And do you remember, T cells do not have antibodies. They have a T cell receptor. It's highly variable and specific like an antibody, but it is not an antibody, right? T cell receptor is not an antibody. B cell receptor is an antibody. And again, do you remember, it's the helper T cells that are CD4 positive, and it's the cytotoxic T cells that are CD8 positive. Do you also remember that epitopes are specific sites on an antigen to which antibodies or T cell receptors bind? And again, do you recall how the natural killer cells work? They recognize your own cells, your nucleated cells, which have internalized their MHC1 in response to some stressor. And once they recognize a cell without MHC1 on its surface, it will destroy that cell by releasing perforins and granzymes to destroy the cell. Concept check number 5. The lymphocyte responsible for recognizing and destroying cancerous cells is the cytotoxic T cells, right? Remember, cytotoxic T cells, they're the ones that, if they recognize foreign antigen on your nucleated cells as presented by MHC1, they will destroy that nucleated cell. And then please note the different types of antibody-antigen interactions. You see all these interactions over here? You should know complement fixation, opsonization, neutralization, antitoxin, precipitation, and agglutination. So for instance, you should know that agglutination is where the antibody aggregates, cross-linking cells or particles into large clumps, right? And then you should know that that's different than precipitation and how that's different from opsonization. So know these different antibody-antigen interactions and their definitions. And then do you remember when you get a vaccination or when you catch an infection? This is known as primary response if you've never had that particular pathogen or vaccination before. And do you remember that you should know that after a little lag period, it's those IgM antibodies that really spike, followed by IgG, right? And then you should know that upon secondary response, it's much, much shorter lag time, and it's IgG that really spikes higher. So remember, this is known as antibody kinetics. So know, again, during the first response, long lag period with IgM first and then IgG. And then during the secondary response, very short lag period with a huge jump in IgG almost right away. And then, of course, please know the difference between active, passive, natural, and artificial immunity. So, for instance, I might say, if you get an infection, what type of immunity is that? You would say natural active. Or if I said mother to child, what kind of immunity is that? You would say natural passive. Or if I said immunotherapy, you would say artificial passive. Or if I said, what is a vaccine? You would say that's a type of artificial active. So be familiar with these terms. And then lastly, we talked about the different types of vaccines. Just be familiar with the different types of vaccines. You should also know, you should know that the Johnson & Johnson COVID vaccine is a viral vector vaccine. While the other ones, remember from Pfizer and Merck, those are RNA vaccines, or also known as mRNA vaccines. And then the final concept check. If only a portion of the pathogen is being used as the basis for a vaccine, it would be called an acellular vaccine. Awesome. So that about wraps up this chapter. Let's head on over after a break time. Let's do break time with Gizmo and Wicket, see what they're up to. We'll be right back with the last two chapters, 18 and 19. All right, welcome back from break time with Gizmo and Wicket. Let's start with chapter 18 now. Remember, chapter 18 kicks off with staphylococci, so we have to learn about these bacterium. You should remember that these guys are salt-loving, so they can inhabit the skin because they are halophiles. They enjoy salt. They're also spherical cells arranged in irregular clusters. They are gram-positives. And remember, one thing I need you to remember is this. They are catalase positive. Remember the catalase test. They are catalase positive. Whereas, do you remember the streptococcus are catalase negative? That's one way of easily telling them apart. You should know that MRSA is a strain of Staph aureus. That's methicillin-resistant Staph aureus. You should know that MRSA or you should know that Staph aureus causes abscesses, which can vary in severity and depth. And do you remember how you can tell Staphylococcus aureus apart from these other staph species listed here? It was the coagulase test. So you should remember that Staph aureus is coagulase positive, while these other staph species are the coagulase negative Staphylococci. And do you recall that Staph aureus is very resistant to penicillin, but MRSA, methicillin-resistant Staph aureus, is even worse because it's also resistant to methicillin. And then vancomycin-resistant Staph aureus is even worse because it's even more resistant. It's resistant to vancomycin. So again, we already touched on this, but which test... All right, so again, we already touched on this, but which test differentiates S. aureus from the other staph species? And remember, it was the coagulase test. And then we talked about streptococci. Remember the genus Streptococci? Remember, these are catalase negative, but usually peroxidase positive. And do you remember they form chains of spherical cells? Whereas, remember, Staphylococcus formed irregular clusters of spherical cells, whereas Streptococcus forms chains of spherical cells. And then do you remember all the Lancefield groups, A, B, C, etc.? So you should know that the group A includes Streptococcus pyogenes, and Streptococcus pyogenes is a big one. You should know that this is the bacteria that causes strep throat, but it can also cause many other issues if it gets into the skin or in your body, but it's most associated, most typically, with strep throat. Remember, if it gets into the fascia, it can form necrotizing fasciitis or flesh-eating disease, which is pretty tough to handle. And then I wanted to touch on the viridans group of Streptococcus because that's kind of important. You should know that this is the group that has the most members that live in the gums, the teeth, the oral cavity, right? So these are typically found in the mouth. And so, for instance, one of the viridans group members, Streptococcus mutans, you should know that this one is involved in formation of tooth plaque, and therefore it's involved with dental caries, which means cavities. And then Streptococcus pneumoniae was so important. Do you remember? It caused about 60 to 70% of all bacterial pneumonias. But it can also cause, remember, you should know that it can also cause otis media, that infection and inflammation and swelling, right? The bulging of the middle ear. And remember, bacterial pneumonia, and remember, Streptococcus pneumoniae is not the only cause of pneumonia. It's just one, it's a very common cause of pneumonia, but it is not the only cause of pneumonia. Even COVID can cause pneumonia. And do you remember this concept check? Bacterial pneumonia by Streptococcus pneumoniae is highly contagious. Now remember, that was false. It's mainly in young or elderly that are somewhat weakened with their immune systems, for the most part. And then do you remember the family Neisseria? We talked about Neisseria. So you should know about Neisseria gonorrhoeae. That is the bacteria that causes gonorrhea. And Neisseria meningitidis, the bacteria most associated with meningitis. But I want you to know this. Just like pneumonia isn't caused by just one bacteria, there's many different bacteria and even viruses that can cause meningitis, because that just means swelling and inflammation of the layers that, wrap around the brain and the spinal cord, right? The meninges. So, again, you should know that Neisseria meningitidis is not the only cause of meningitis, but it's a common cause of meningitis. And it's a common cause in young adults. Oh, and we just touched on this. A common cause of meningitis in young adults is found in the genus Neisseria. Remember, Neisseria meningitidis, but it's not the only cause. All right, and that leads to the end of chapter 18. We just got one more chapter to do, so let's do it. All right, chapter 19. Now we're talking about the gram-positive bacilli of medical importance. You should definitely know that endospores are formed by members of the genus Bacillus and the genus Clostridium. And do you remember that the bacteria Bacillus anthracis, again, it's an endospore forming bacterium that causes anthrax. Whereas, do you remember Bacillus cereus? Do you remember that one? You should know that this one is associated with food poisoning. You should remember that Clostridium perfringens causes gas gangrene. So remember this, we just touched on this. Which of the following is the mode of transmission for Bacillus cereus? Remember, it's ingestion, right? Because it's so closely related to food poisoning. This is why we need to refrigerate our food. And then do you remember tetanus is caused by a bacterium called Clostridium tetani, And you should know this. You should know that there is a vaccine for tetanus, right? You've probably had it, right? So you should know that there is a vaccine for tetanus. In fact, there's also a vaccine for anthrax, Bacillus anthracis, though it's mainly, remember, given to soldiers, right? The military, right? Because they're the most at risk of encountering it in a negative way. And do you remember that tetanus causes, contraction of all the muscles, right? That causes uncontrollable contraction. And then C. diff, let's see if you can beat Wicket. Okay. Do you remember what treatment does C. diff infection usually follow? Like, what do they have in common? What makes you at risk of C. diff infection? Can you beat Wicket? That's right. Wicked as always. Antibiotics, right? It's patients in a hospital. They take a round of antibiotics. Unfortunately, that destroys their gut flora. And then that C. diff or Clostridium difficile is free to flourish and take over the intestines. So you often see it following antibiotic treatments. And it's picked up at the hospital a lot of times, unfortunately. And do you remember that C. diff causes colitis, right? Inflammation and bleeding in the colon. And then do you remember Clostridium botulinum? This is the bacterium that gives you botulism, and it's found in home canned foods often. You should know that botulism toxin, not only does it paralyze you, but it acts on the neuromuscular junctions. Remember that? So we just mentioned this. The action of the botulinum toxin is on what? It's on the neuromuscular junctions. Next, we talked about listeria. Remember, these can survive in cool environments. Remember how listeria works? Isn't that interesting how it can be, it stimulates its own phagocytosis by macrophages, but then it lives inside of the macrophages. It's kind of interesting. And then we learned about a very important bacterium called Corynebacterium diphtheriae. Remember, this is the bacteria that causes diphtheria, the disease. But remember, this is a respiratory disease. You remember that? And there's a vaccine. Remember part of that Tdap vaccine? In fact, it's the same vaccine series, the same mixture as the tetanus vaccine and DAP. The D in DAP stands for diphtheria, right? So you should know there is a vaccine for diphtheria, Corynebacteria diphtheriae. You should know there's a vaccine for tetanus, Clostridium tetani. And do you remember Propionibacterium acnes is the cause of acne? Corynebacterium diphtheriae causes pseudomembrane and respiratory distress and can be prevented with a vaccine. That is true. You should remember that. Then we talked about the Mycobacterium. Remember, these are bacteria that are called acid fast. These are the acid fast bacteria because they have this waxy structure in their cell wall called mycolic acids. And the one we talked about in detail was Mycobacterium tuberculosis. And do you remember that this tuberculosis is a lung disease usually, and it's spread by respiratory droplets, right? So if you cough, you sneeze, you exhale and you release respiratory droplets. That's how Mycobacterium tuberculosis spreads. And remember, only about 5 to 10% of infected people actually develop the TB disease, the clinical disease. So remember, we touched on this. Tuberculosis is spread via respiratory droplets, right? And then we talked about Mycobacterium leprae. You should know that this one is the cause of leprosy. And there is no actual vaccine against Mycobacterium leprae. This is one that we do not have a vaccine for. And then lastly, we mentioned the actinomycetes. You should know that these are filamentous bacilli. They have a filamentous cell shape. So again, these are known for being filamentous bacilli. And the actinomycetes, like Actinomycetes israelii, is responsible for actinomycosis, while Nocardia brasiliensis causes pulmonary disease called nocardiosis. And that's it. We've reached the end. Again, please study everything, for the exam. I do my best to prepare you for the exams with these reviews, but I may not remember every little thing, but I try to stress those super important things to know. And let's move on to exam board material.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.